Good day, everyone. In this short teaching session, we are going to discuss physiological effects of catecholamines. I will explain few important aspects related to physiology of catecholamines, as indicated by the list of learning objectives for this session. After this presentation, you will be able to describe the catecholamine secretion by the adrenal gland, to describe the subtypes of adrenergic receptors and their intracellular transduction mechanisms, and identify physiological effects induced upon stimulation of different subtypes of adrenergic receptors. Let's start with explaining some introductory information. The term catecholamines applies to adrenaline and noradrenaline, and if you look at their chemical structure, you will find the catecholamine and the amino group, and this is why these agents are called catecholamines. The catecholamines are secreted by the adrenal gland, which is a small endocrine gland localized bilaterally in the abdominal cavity just above the kidney. The adrenal gland has two important structural elements. This is adrenal cortex and the adrenal medulla. The adrenal medulla is made of chromaffin cells, and these cells secrete catecholamines. The main factor which promotes secretion of catecholamines is sympathetic stimulation, and this is because the chromaffin cells receive inputs from preganglionic sympathetic fibers that originate in the spinal cord. Upon stimulation, these fibers release acetylcholine, which is acting as a sympathetic neurotransmitter in this location. And acetylcholine then stimulates nicotinic cholinergic receptor on the chromaffin cell, which promotes exocytosis of granules containing adrenaline and noradrenaline. Subsequently, the catecholamines diffuse into the bloodstream, and they're distributed with the blood throughout the body and produce their effects on the target tissues. So overall, these agents are acting like hormones because they're secreted by the endocrine gland and released into the blood and produce their effects at the organs localized at a distance from the site of their production. Once catecholamines arrive to their target organs, they produce effects by stimulating plasma membrane receptors subdivided into two types: alpha adrenergic receptors and beta adrenergic receptors. Both receptor types are G protein coupled receptors, but they are different in terms of intracellular enzymes which are targeted upon receptor activation and the second messengers produced in the cell. The beta adrenergic receptor is coupled to adenylate cyclase, which generates cyclic AMP, and this is acting as the second messenger, which activates another enzyme within the cell, such as protein kinase A, and the PKA then phosphorylates cellular proteins, which induces physiological response. The alpha adrenergic receptor is coupled via G protein to another enzyme, phospholipase C, which generates two second messengers, such as inositol trisphosphate and diacylglycerol. Inositol trisphosphate then stimulates calcium release from endoplasmic reticulum, and diacylglycerol stimulates protein kinase C. So in this case, calcium and protein kinase C are intracellular effectors which are targeted upon alpha adrenergic receptor stimulation. Beta adrenergic receptors are subdivided into three subtypes: beta 1, beta 2, and beta 3. These subtypes have different localization in our body, and they mediate different effects. The beta 1 receptors are mostly concentrated in the heart, and they mediate cardiostimulatory effects of catecholamines, such as increase in heart rate and increase in cardiac contractility. Beta 2 receptors are present in few important locations. First, they are found in the smooth muscle, which could be bronchial smooth muscle or uterine smooth muscle or vascular smooth muscle. And the activation of beta 2 adrenergic receptor is inducing relaxation of the smooth muscle, which translates to bronchodilation, inhibition of the uterine contractions during parturition, and vasodilation. In addition, the beta-2 adrenergic receptors are found in liver, in the skeletal muscle as well, where they mediate some important metabolic effects. In particular, in liver, stimulation of these receptor subtypes contributes to breakdown of glycogen and stimulates synthesis of glucose from amino acids. This is called gluconeogenesis. Therefore, a great amount of glucose is then released from liver to the blood. In the skeletal muscle, stimulation of these receptors reduces glucose uptake from the blood. So all these actions taken together contribute to increased blood glucose levels, which is called hyperglycemia. Finally, there is beta-3 adrenergic receptor subtype, which is found in adipose tissue, and its stimulation contributes to lipolysis, which releases free amino acids, free fatty acids into the blood, and subsequently the free fatty acids are oxidized to generate energy. So they're used as metabolic fuel. Now, what about alpha-adrenergic receptors? These receptors are subdivided into two types, alpha-1 and alpha-2. The alpha-1 receptors are found in our body in three important locations. First, they're present in the vascular smooth muscle, where their stimulation produces vasoconstriction, resulting in increased blood pressure. Secondly, alpha-1 adrenergic receptors are also present in the circular smooth muscle that forms sphincters in the gastrointestinal tract and the urinary bladder. And here, these receptor stimulation contributes to constrictional sphincters, which inhibits gastrointestinal peristalsis and bladder empty. These receptors are also found in the radial muscle of iris in the eye, and upon stimulation, they lead to dilation of eye pupil, which is called mydriasis. Finally, we also have alpha-2 adrenergic receptors, and these receptors are localized on presynaptic terminals of the adrenergic neurons, where they control release of noradrenaline. Upon stimulation, the adrenergic neuron is releasing noradrenaline into the synaptic cleft, and noradrenaline is then producing effect on the target tissue. But in the meantime, noradrenaline also stimulates alpha-2 adrenergic receptor at the presynaptic terminal, which inhibits subsequent release of noradrenaline. So this means that alpha-2 adrenergic receptor is involved into the negative feedback mechanism, which controls release of noradrenaline at the presynaptic level. And this mechanism ensures that there is no excessive release of noradrenaline upon stimulation of adrenergic neuron, which would otherwise lead to depletion of noradrenaline stores in the presynaptic terminals. Now, let's briefly summarize what you learned from this session. Now you know that catecholamines are secreted from the chromaffin cells in the adrenal medulla. You also learned that effects of catecholamines are mediated through activation of alpha and beta adrenergic receptors. And in particular, alpha adrenergic receptors mediate effects of catecholamines on the smooth muscle and the presynaptic release of noradrenaline from the adrenergic neuron, whereas beta adrenergic receptors mediate action of catecholamines on the heart, the smooth muscle, liver, and adipose tissue. My final slide, just to highlight some important learning resources. I highly recommend these two review articles, which were recently published, and now they are available in PubMed. These are the links. And if you have any questions related to this topic, you are very welcome to contact me by sending email. Thank you for listening.